Welcome, everybody, to this American Society for Cellular and Computational Toxicology webinar. I hope you will enjoy today's program. You can see on your screen that we have a website where you can learn a lot more information about us. If you're not a member of the Society, please consider becoming a member. You get access to recordings of the webinars as well as other benefits. And my name is Christy Sullivan. I'm the secretary of the ASCCT. And I just want to briefly mention that we have an annual meeting. This is a picture of our last annual meeting, which was in September in Research Triangle Park, North Carolina. It was a very great meeting. We had lots of presentations, a panel discussion. We had a poster session, and we gave out some awards to students. And in fact, that's what we're talking about here today. We will have our next annual meeting for 2017 is our 6th meeting, and it will be in September again, but this time it will be in Washington, D.C. We haven't yet set the dates, but information will be going out once we do. And if you would like to be on the program committee and are interested in helping to plan the meeting, please feel free to email me at ksullivan@ascctalks.org. And so I'm going to go ahead and get started. At our last meeting, we awarded 2 students for their work, and we're happy to have them here today to describe their research. Our first presenter is Emma Bowers. She is a PhD candidate at the University of North Carolina in the curriculum in toxicology. She's working at the US EPA Human Studies Facility in Chapel Hill, North Carolina. Emma is the second winner of the Ed Carney Award for Predictive Toxicology, which is an award that the Society gives out every year to young scientists. And she will present her work on ozone exposure in vitro. So Emma, I'm going to go ahead and give you control.
Okay, can you see my screen?
Yes.
Okay, excellent. All right, let me just minimize here. Okay. Can you see the GoToWebinar window, or is that just on mine?
No, we can't.
Oh, okay, good. I just want to make sure that it's not obstructing everyone's view. So, okay, thank you so much for allowing me this opportunity to present my research. And today I'm going to be talking about my dissertation research in which I'm exploring ozone response heterogeneity and adaptation, which are really complex in vivo responses that are really not well understood. Hence, we're really trying to recapitulate these things in vitro. And so I'd like to just start by really discussing air pollution. Air pollution is a really important public health issue. So 1 in 8 of total global deaths are actually the result of air pollutant exposure. And that's led the World Health Organization to find that it's the world's largest single environmental health risk. And if you look over to the right of the slide, you can see that many of the deaths that are attributed to ambient air pollution really are related to things like ischemic heart disease, stroke, and COPD, so cardiopulmonary diseases. And so that sort of begs the question, you know, how is it that air pollutant exposure really causes these deaths? And the answer is quite complex, but really one of the key things that we're learning is that air pollutant exposure causes inflammation, which can really cause the development of these diseases or sort of trigger acute lethal events. And because of this, it's really important that we try to understand the relationship between air pollutant exposure and inflammation. And to do that, one of the model oxidant pollutants that's commonly used is ozone. So ozone, unlike many other air pollutants, it's not directly emitted. In fact, it's actually formed in secondary photochemical reactions between nitrogen oxides and VOCs. Ozone as a model pollutant has been studied for decades, and importantly in this case, it's really well established that it can cause pulmonary inflammation. And this next slide is really demonstrating that point. Here you have the average neutrophil or PMN infiltration into the lungs of individuals who've been exposed to clean air and ozone. And this is just showing you the average neutrophil infiltration here. And you can see it's a moderate uptick. But that's not really the interesting thing here. The interesting thing is if you look at what happens to these individuals who experience ozone exposure. And you can see really that you have marked heterogeneity in response. And I just wanted to really highlight the fact that these are healthy individuals. And you can see some of the responses in these healthy individuals can actually potentially exceed the responses that you might see in traditional risk groups, so those who have pre-existing conditions, who might be elderly, etc. And so really this is demonstrating that we really need to understand sort of the basis of this inter-individual variability, which unfortunately we really don't right now. And so sort of continuing on this sort of trend with discussing inflammation, there's another response that's also poorly understood but very important for us to understand, and that's ozone adaptation. So here this is showing similar data from a clinical study where you have neutrophil and lymphocyte infiltration into the lungs, and you can see what happens after 1 day of exposure and then after 4 days of exposure. You can see there's a decline, and the decline is actually mirrored by similar declines in cytokines present in the lungs, so cytokines like IL-8 and IL-6. So really what you're seeing is that with repeated ozone exposure, you have less of a pro-inflammatory response. And again, this has been observed in epidemiological studies, clinical studies, animal studies for a really long time, but we have little to no information as to why it happens or why, what the health implications are. So I've told you over and over again, we don't understand these things. Well, why don't we understand them? So first of all, these things are really explored often using human clinical studies, and as we all know, these can be inefficient. You're kind of limited to sort of how you can manipulate the system. Really, you're mostly observational, and of course, if you're anytime you're doing sort of a repeated exposure situation, you have safety limitations. You have many of the same issues with animal studies. But then, of course, because you have a limited number of strains that you can study, you have an issue of can you really, effectively study heterogeneity? And of course, there's always the question of does this really mimic human biology? So an ideal way to study these things would be using an in vitro model. But unfortunately, if you use a cell line, that's really an N of 1 if you're trying to study heterogeneity. And unfortunately, if you're trying to study ozone exposure over multiple days, cell lines don't exhibit contact inhibition, so they have a tendency to overgrow. And so that's a huge confounding factor if you're trying to study adaptation. So these are the problems. So why do we need to overcome these problems, right? If we understand heterogeneity, first of all, then perhaps this can help us predict susceptible populations. Second, if we start to understand the mechanisms of this response, heterogeneity, and adaptation, perhaps we can start to leverage these things to mitigate exposure effects, either through behavioral changes or perhaps medical intervention. And finally, we really need to start understanding, is adaptation good or is it bad? We are used to hearing about how inflammation is a bad thing, but that's not necessarily a given. So all of these things being said, we really sort of need to move forward on these issues. So as a solution for this, we decided to try to recapitulate these things using an in vitro model. And to do that, we use primary bronchial human epithelial cells. So these cells are collected from human donors, plated on transwell inserts, and then they're taken to an air-liquid interface. The cells are allowed to differentiate over 24 days, and after that period, they're exposed to ozone on a daily basis. And again, these cells exhibit contact inhibition. At this point, they're relatively stable. There's not issues with overgrowth. They also really do a very good job of mimicking physiologically relevant responses. So in the middle of the slide, you can see the exposure diagram here that I've used, and you can see that we've done 4 days of exposures. As a control, we have 4 days of filtered air. To look at acute exposure, so a single ozone exposure, we have 3 days of filtered air and then 1 day of ozone exposure on day 4. And then to study repeated ozone exposure, these cells receive ozone each day, again, for the 2 hours on every day of exposure. And so we only harvest on day 4 because we're normalizing for the number of days in culture as well as any sort of cell handling. So on day 4, when we harvest, we look at protein, and we're also looking at a couple other endpoints, which I'll discuss. But some of our main endpoints are looking at these genes, which are on the bottom of the slide. These are ozone-responsive genes, and genes that are also known to be really important in determining the pro-inflammatory response, and as well as balancing sort of oxidative stress. And so when we look at the expression of these genes, here I'm showing you the expression from 13 different cell donors. So again, 13 different individuals from which cells were collected. You can see that indeed we have response heterogeneity. And really, if I'll guide your attention up here to IL-8, you can see that it's interesting because these donors almost segregate into 2 different groups. We have sort of a low responding and a higher responding group. And you'll notice that these donors up here have hollow circles. And I've done that because I wanted to show you that if you track these donors through the rest of these genes, noted again with the hollow circles, you can see that if you're a high responder for IL-8, chances are you're also going to be a high responder in these other genes. And so now if we look at repeated ozone exposure, you can see that we indeed have something that appears to be in vitro adaptation. So the mean expression of these genes has declined. But interestingly, this really seems to be driven by a part of these donors. So the donors that are up here that are sort of the high responders are then the ones that are adapting and having reduced expression, whereas the donors that begin low sort of stay low and consistent. And so this really sort of boils down to that we almost have these 2 ozone response phenotypes, which we sort of call high and low responders. And so from here on out, this is going to be important when I start looking at some of the mechanisms associated with responsiveness. And so just know that when I refer to high responder, it's donors that have IL-8 expression above the mean. And again, we use IL-8 because it's really important for pulmonary inflammation, and it's sort of an indicator of overall inflammatory response. And again, the low responders are below the mean, which is about 4.5. So you can imagine that, we're seeing this response heterogeneity, but then you have to wonder, okay, the response phenotypes that you're seeing here, is this somehow due to, an effect of what's happening that day in lab? Maybe it's a confounder with how these cells are plated. Is this stochastic? So we really wanted to see if this ozone responsiveness was an inherent property of these cells. So then we looked at some exposures for which we had, some interesting data. So first of all, we looked at cellular responses in which we had cells from a single donor that were collected at essentially the same time, but were thawed and plated and exposed to ozone at different times. And this is going to give us a sense of whether the cellular responses are sort of reproducible. Then what we also wanted to do is look at, okay, if we take one individual and then do a bronchoscopy and collect these cells at different periods, so say at one point in time and then, several months later, for example, at another point in time, then thaw and plate these cells and expose them to ozone, how stable is this response over time? And what we've done basically is plot the IL-8 response from the first exposure or first bronchoscopy exposure on the x-axis against the second exposure on the y. And what you can see here is that so far it's definitely looking like this ozone responsiveness is sort of not stochastic. It's not. It's actually quite reproducible, and it's really an inherent property of these cells and donors, which is pretty interesting. So now feeling kind of confident in our model system and feeling like, this responsiveness is, again, an inherent property of these cells, we felt like we could move forward and start to investigate some of the mechanisms that could be influencing ozone responsiveness. And so whenever I've discussed adaptation in the past, I've often been faced with sort of this pre-existing hypothesis that's been floating around the literature for many years. And that is that ozone adaptation could be the result of increased antioxidant capacity. And really what this is summarizing is the fact that during a single ozone exposure, you could have an increase in reactive oxygen species, which could then really flood the cell and cause this pro-inflammatory response. However, upon repeated exposure, the cell has the opportunity to sort of ramp up its antioxidant response and sort of restore balance to the system, and therefore you don't really have as much of that pro-inflammatory response. And there's sort of been these two sub-hypotheses that we wanted to address. The first is that you could have the upregulation of oxidative stress response genes. And the second is that this could also happen due to the antioxidant capacity of cellular secretions. So we briefly looked at these two aspects of antioxidant capacity. So first what I'm showing you is a gene expression from a PCR array in which we looked at about 80 genes that are known to be involved in oxidative stress response. And this is essentially gene expression that's from a single ozone exposure that's normalized to filtered air. It's from 6 donors, which are noted on the left, and they're ranked by IL-8 response, so a 3.25-fold change on the top, about a 9.26-fold change on the bottom. And then what I'll do is juxtapose this with the repeated ozone exposure. And what you're really looking for, if this hypothesis was sort of true, you'd be looking for red here, right, indicating an upregulation during repeated ozone exposure. But as you can see, we're seeing mostly gray and blue, indicating that there's either no change or a very slight downregulation. We did do a statistical analysis to see if there were any genes that were differentially regulated between single and repeated ozone exposure, and those are indicated by the black arrows here up at the top, but all of these genes were actually very slightly downregulated. So again, this is showing that there are some differences between single and repeated ozone exposure, but it's not sort of an upregulation of these oxidative stress genes. So next we looked at the apical secretions. And to do that we used an oxygen radical antioxidant capacity assay. And this assay is based on the fact that you basically take a fluorophore and add a radical to it and then monitor fluorescence over time. If your sample has an antioxidant in it, it will sort of prevent the degradation of that fluorophore, and then you can do a calculation where you can basically look at the area under the curve between a blank and a sample that has, again, antioxidant in it. And so we did that with cell secretions that were collected from cells that were exposed to 3 days of filtered air or 3 days of ozone. And those are indicated sort of comparing the black and the gray bars, and we did that for 6 donors. Again, they're sort of ranked in their IL-8 response. And really the take-home here is that you're either not seeing any difference or you're actually seeing a reduction with ozone exposure. So again, that's sort of negative data showing that we're not seeing, and it basically showing that an increase in the antioxidant secretion of these cells is not really going to explain ozone adaptation. So basically, I just showed you a bunch of negative data. But don't be disheartened. I'm about to show you something exciting now. So we then moved on to something that we felt like we were sort of more excited about, that we felt like was probably going to be the driving mechanism. And a lot of this groundwork was really done by Sean McCullough. And he demonstrated that in primary bronchial epithelial cells, the MAP kinases, P38 and ERK, are really essential for ozone pro-inflammatory signaling. And he showed this in basically cells that were not fully differentiated, they were partially differentiated. And really the paradigm as it stands is that, you have ozone exposure that generates reactive oxygen species, and then these sort of activate EGFR and other kinases, eventually sort of converging on ERK and P38. These then activate transcription factors and effector proteins, which cause the transcription and translation of inflammatory genes. To sort of reinforce the point that these are really necessary in my system, I use two inhibitors of ERK and P38 shown here to see if, okay, during ozone exposure, if we inhibit these kinases, are we going to see expression of pro-inflammatory genes? And this is really just showing you that data where, again, if you have in high responders, you can see you have an ozone vehicle iolate induction here around 8 to 10 fold. But then if you use the ERK and P38 inhibitors, you see a reduction. But really you only see a significant reduction once you use the ERK and the P38 inhibitor together. And so really, again, this is just reinforcing the fact that ERK and P38 are really necessary for pro-inflammatory gene expression. And so this sort of led to the hypothesis, could the difference in ozone responsiveness really have to do with the sort of extent of activation of these pathways? So what I then did was I looked at the activation of these pathways in both high responders and low responders. And again, just to remind you, the high responders have sort of that iolate that's above the mean, low responders are below the mean. And you can see ERK here in the higher responders, we are activated, not so much in the low responders, and this is paralleled in MEC and RISC, which are above and below ERK. And over here you can see densitometry analysis, and this is for a total of 12 donors, 6 high and 6 low responders, and you can see that essentially we have ERK activation and the activation of the kinases upstream and downstream of ERK. However, if we then look at P38 activation, here I'm showing you P38 and its upstream kinase MKK4, you see we have some slight activation of these things, but really there's no difference in the high and low responders. So really this sort of leads us to believe that responsiveness is really tied to the extent of ERK activation, not necessarily P38. So what about adaptation? So if we look at these two kinases during repeated ozone exposure, what you can see is that you have an activation of ERK, but then actually a decline during repeated ozone exposure shown here. And sort of this really parallels the adaptive response and really suggests that this could be one of the central mediators of ozone adaptation. And again, you're not really seeing any association with P38 activation, although we do see some slight activation. So in conclusion, I just wanted to reiterate that using this model, we have these two ozone response phenotypes, which I think are really interesting. We have high responders, which have a robust pro-inflammatory response and an adaptive response. And then we have low responders, which have either a minimal pro-inflammatory response in which adaptation is either mild or undetected. And what we can do now is use this model to try to understand the factors that are really going to differentiate high and low responders. And furthermore, we can make some inferences regarding the health effects of ozone responsiveness and adaptation. And just to sort of put my 2 cents in on the sort of health effects of ozone adaptation, since it's been debated for a long time, I would argue that perhaps we are being a little too naive as to try to classify it simply as good and bad. Instead, I think what we need to do is start thinking about it in terms of subpopulations. For example, if you have cardiovascular disease in which, inflammation could really lead to the exacerbation of that disease, perhaps adaptation could be a good thing for you. However, if you're somebody where you're immunocompromised and you get exposed to ozone repeatedly and your immune system is dampened, perhaps that can be really bad for you. So with that, I would just like to acknowledge ASCCT again for this opportunity. My mentors, David Diaz-Sanchez and Sean McCullough, my committee, my funding, and with that, I'd like to take any questions.
Great. Well, thank you very much, Emma. And we're going to go ahead and unmute everybody to see if we have any questions. The floor is I had a for you. When you were talking about your exposure protocol, you had a 4-day exposure protocol. Can you talk a little bit about why you decided to do that? Can hear me. Did you hear my question?
Hello? Sorry,
Oh,
yeah, I heard there was a lot of noise, so I had trouble hearing your question.
I'm sorry about that. Okay, so I just was wondering, when you did your exposure protocol, you had a 4-day exposure for the cells, and was wondering what was behind that, why you chose that 4 days.
Yeah, so basically from reading the literature, it seemed as though in individuals who had exhibited inflammatory ozone adaptation, normally in human studies, data points were collected after 4 days of exposure, and that's when adaptation was visible. And so we sort of wanted to just reflect that same sort of time course.
Okay. All right. If there's another question for Emma, then why don't you please write your question in the question feature. You'll see on the control panel there's a question feature. We're going to, oh, Ray. Let me see if I can see your question. Ray Tice has a question. It says, ozone seems to be especially harmful to older people. Is there an effort to look into this in vitro?
So that's an interesting question. So we're seeing that these cells, the responsiveness of these cells is an inherent property that has to do with the individuals they come from, right? So I think it could be interesting to see if we collect primary cells from older individuals, is that going to be reflected in sort of the responsiveness of those cells? And so that's certainly something that we could explore in the future. And so, yeah, I think that would be really interesting.
Great. I'm just going to check and make sure no one's got their hand raised. Okay. I I apologize, folks. So there are two ways when you're muted to ask a question. You can raise your hand, and then I can unmute you individually, or you can write a question in the question box. Okay, so we're going to move on to our next presenter. Thank you again, Ellen. Thank you again, Emma. And now we're going to move on to Ellen. So our next presenter is Ellen Garcia. And Ellen is a PhD student in the Department of Biological Sciences at Virginia Tech. Ellen won the first Tox 21 Student Award, which is an annual award established by a generous donation by our member Ray Tice. And she will be presenting on silver nanoparticle exposure in vitro. So let me go ahead and give you the controls.
Okay, can everyone hear me now? Sorry, I seem to be muted.
I unmuted you.
And can everyone see my screen okay?
Yes.
Wonderful. Okay. Well, thank you, Christy, for inviting me to do this webinar. It's the first time that I've done anything like this, so it is feeling a little weird, but I'm excited to talk about my work with everyone listening in. So as it was said, my name is Ellen Garcia. I'm a PhD student working at Virginia Tech. I work in Daniela Cimini's lab, which is a lab really focused on understanding cell division. And I came into being interested in nanoparticles and nanotechnology kind of just through serendipitous events of my graduate work and meeting the right people. And through all of that, I kind of designed a project to work on as a PhD student that's really focused on alternative animal testing techniques and experimentation. I really wanted to be able to apply my dissertation research to alternative animal experimentation in academia and throughout industry. And so that's kind of what I'm going to be talking to you about today is one project that makes up kind of my whole dissertation with that real main theme of alternative animal testing technique. So today I'll be talking about one main research project, and let me make sure I can figure out how to change slides. Okay. So my main research objective with this project is to develop and validate a cell division-based toxicology approach by investigating the mechanism of toxicity from silver nanoparticles on human retinal pigment epithelial cells. Now I know this objective is kind of a mouthful, so I'm just going to walk you guys through various parts of the objective. The first thing I'm going to focus in on is why I have chosen to focus my toxicology approach on cell division. So the act of one cell dividing into two is a very fundamental and conserved process, and it's happening millions of times in your body right now, and this process is fundamental for organisms to develop and to sustain. On the screen here I have a diagram on the left showing the cell cycle, and I'm really interested in this red portion here, which is indicating that stage of mitosis and cytokinesis. And what we really wanted to look at is if you perturb the system of mitosis or cell division, is that a better indicator of stress or of toxicity than just looking at cell death, for example. And we feel that this is a very good system to understand how chemical exposure affects cell populations. Secondly, the reason I am focusing on silver nanoparticles kind of as my chemical of interest for chemical exposure really has to do with the fact that silver nanoparticles are one of the most widely employed nanotechnologies in kind of our working society right now. I think it's right below titanium oxide nanoparticles as far as the amount that's being put out in consumer products and medical products. So for example, you can find silver nanoparticles in a wide range of textiles and medical products such as catheters, wound dressings, and the list kind of goes on and on. So it's a very popular nano material that's been used, but we don't really understand the mechanism of toxicity to human cells, to non-bacterial organisms. And so with that, I've decided to kind of attempt to contribute more mechanistic understanding of how silver nanoparticles interact with human cells. And just to draw your attention to the center of the screen, I do have a representative TEM image that I took of the nanoparticles I'm working with to show you relatively their size and shape. Next, the reason I am using the model of human retinal pigment epithelial cells is for a few different reasons. One main reason is in the literature for silver nanoparticle toxicity, there's really no evidence of how silver nanoparticles interact in the eye, and therefore I liked the idea of working with an eye-related cell. And just to bring you to the diagram here on the right, the retinal pigment epithelial cells, the RPE cells, are the very back lining of your retina. And as an interesting side note, these cells have the highest level of, or the highest rate of phagocytosis in the whole body. Another reason why I appreciate using this model to understand nanoparticle exposure, besides that retinal cells are lacking from the literature, is I think using epithelial cells is a really important aspect as well. When you look through the silver nanoparticle toxicology research literature, there's a lot of evidence in fibroblasts or cancer cells, and we feel that using an epithelial cell model will highlight a lot more relevant information for actual exposure in the body. So before I explain my experimental design for silver nanoparticle exposure, I first want to just quickly show you size characterization of my nanoparticles. I'll also tell you a little bit more about them. So I purchased them from a commercial manufacturer in the form of powder. I decided to put them into liquid suspension in order to best expose my cells in liquid culture. And these nanoparticles are coated in PVP as a stabilizer, and they are reported by the manufacturer to be at 20 nanometers. And when I run dynamic light scattering analysis, I see a range of sizes, but all relatively staying in the nanometer size range. So I am working with a heterogeneous size population of silver nanoparticles, and that's further shown to you in these TEM images here on the right-hand side. So with these nanoparticles, once I put them into liquid suspension, I then laid out an experimental plan in order to better understand how my cells respond to the nanoparticles. So I'm going to walk you through this diagram, and really all of my experimentation is very based in this flow, and so I want to make sure that it's very clear what I did, so in a little while when I'm referring to my data, everyone will be on the same page. So as with all in vitro work, I had to culture my cells. I would seed them at specific densities and give them 24 hours to adhere in culture and kind of become happy after being passaged. After one day, I would then treat my cells with silver nanoparticles, and I used an array of concentrations of which you'll see throughout my data, ranging all the way from 0.5 micrograms per milliliter all the way up to 100 micrograms per milliliter. And so I would treat my cells for 24 hours. They would incubate in the nanoparticles, of which during I would conduct live cell imaging. After 24 hours of incubation, the cells would then either be harvested for fixed cell analysis, or they would be washed out, and therefore the nanoparticles would be taken out of culture. If the cells were harvested for fixed cell analysis, I would be able to look at various types of quantifications. So just for example, I could find out what the mitotic index of the cell population was, what potentially the mitotic stage distribution looks like, and I can also look for mitotic defects or interphase defects. I also should point out that information that I gather when I conduct live cell imaging is really where the single cell analysis comes from. I'm able to look at single cell behaviors and the timings of those behaviors using the live cell imaging. Now, doing one treatment, one kind of section of this diagram here is what I considered an acute exposure or an acute treatment, so only one 24-hour treatment. Now, in order to model more chronic exposure to silver nanoparticles, I continued these cell populations in culture and would treat them more and more times. So that's the second option that I spoke about, that if the cells are washed out, I give them a few days, 3 to 4 days to recover, and then after the cell populations have recovered, I'll do this whole cycle again, and they'll be treated again for 24 hours, washed out, and allowed to recover and passaged. And so when I did this cycle for 6 different times, 6 different silver nanoparticle treatments, I'm considering that moderate exposure. And when I did 12 total treatments, and that's 12 different individual treatments on the cell population, I considered that chronic exposure. So those three kind of labels I'll be using for the rest of my data presentation. And just to note, this entire cycle, this entire long-term experiment lasted about 6 weeks. So it was a total of approximately 2 treatments per week. Something else I would also like to mention that I don't have a lot of data in this presentation about is I did do negative controls with cells that were not exposed to nanoparticles, so you will see that control data, but then I also did 2 positive controls where I treated cells with silver nitrate to see the effect of silver ions and distinguish that from kind of a nanoparticle effect. And then I also did a positive control using just PVP exposure, which, as I said a little while ago, was the coating that stabilizes my nanoparticles. So the first video I'd like to show you is a phase contrast video. This is from control RPE cells. And what you're really noticing here is that cells look healthy. Their morphology looks normal, their behavior, their movement looks normal, and you're seeing cells continually round up and finish mitosis, which is the real kind of action you're seeing with all of these cells dividing into 2. Now, when I show you a video of treated cells, the story looks a lot different. You can tell that the cells are stressed, you can tell that they're unhappy based on their morphology, their movement, and you're also seeing a lot of cells round up and remain rounded up and not completing division. And so these cells here, I'll play this video a few more times, are the cells that round up and they stay rounded up for a long length of time. And so these cells we consider to be arrested in mitosis. Now, when you quantify this in the live cell imaging, what you see is that there is a concentration-dependent decrease in the amount of cells that complete mitosis and an increase in these rounded up cells, or cells that we consider arrested in mitosis. Now, when I look further into these rounded up cells, these cells that just kind of remain in this rounded up phenotype, with fluorescent microscopy, you see that these rounded up cells don't have a characteristic microtubule kind of cytoskeleton structure, and you also see that their DNA is condensed and not in order. And so when we take these rounded up cells and we harvest just those rounded up cells and we analyze them using, I did an experiment where I stained them with trypan blue, and that will, if the cells are dead, the trypan blue will enter the cells. If they're alive, they won't. You see a concentration-dependent decrease in the cells that are viable, that are still alive, and an increase in the amount of cells that are dead. So of these cells that we're observing in our live cell imaging that are rounding up, most likely, most of them are on the path to dying or are already dead. Now, another interesting phenomenon we were able to see from the live cell imaging is we were able to observe a physical interaction with the cells and the nanoparticles in cell culture. So if you really watch this leading edge of the cells, you really see them engulfing the nanoparticles and just kind of gobbling them up. And there are some proposed mechanisms of why and how this is happening, but when you quantify it, 100% of the cells, including the cells that go on and divide, finish division, engulf the nanoparticles. We can also verify this when you look at fluorescent microscopy. So on the left-hand side, I'm showing you an image of control RPE cells stained for their DNA and their microtubules. Then when you look at the same staining pattern in treated cells, you see this very obvious contrast of what appears to be black inside of the cells. And you can see that in cells that are in interphase and cells that are in mitosis. So these black dots that the arrows are pointing at indicate internalized silver nanoparticles. Something I won't be showing you today is the fact that we have, or we are attempting to do one more quantification for internalization, and we're going to be doing some electron microscopy to further confirm that the nanoparticles are inside the cells. Next piece of data that I'd like to show you is that we were able to see a concentration-dependent increase in the time it takes for cells to complete mitosis. So of the cells that do complete it, the length of time that it takes them to move through all the stages of mitosis increases. And we do see that, this becomes significant with higher concentrations of silver nanoparticles. Now, I also gathered timing information from cells that were washed out of the silver nanoparticles, so cells that were in the recovery phase of treatment. And here on the left-hand side, I show you the timing of cells going through mitosis 24 hours after being incubated in silver nanoparticles. And this graph is after one treatment, an acute treatment, and the right-hand graph is showing you the timings of cells when they're recovering after having gone through chronic treatment. And what's interesting is you do see that the cells recover their mitotic timing, and we are still looking into kind of why this is happening in the cell populations. Now, the most kind of interesting result that we found, I know this slide is kind of busy, but I'll work you guys through all of it. One of the more interesting things that we found is we do see a concentration-dependent increase in the amount of binucleate cells in the population. We see a concentration-dependent increase and a time-dependent increase. So when you go from acute to moderate to chronic exposure, you're seeing more and more cells that are binucleate. And the same goes when you look at lower concentrations compared to high concentrations. Right here on the right-hand side, I show a fluorescent image of a binucleate cell. We also see this kind of underlying population of multinucleated cells, which we are further looking into quantifying. We're not sure if they're actually multinucleated, or I showed some examples down here of just some really funky nuclear phenotypes that we come across in treated cells. So multinucleated cells might actually more be like highly lobed, or we're seeing very odd-shaped nuclei. And so we're further looking into better quantifying these other underlying phenotypes that arise after silver nanoparticle exposure. But the take-home message is that a percent of cells, instead of dying from silver nanoparticle exposure, they do end up becoming binucleate. So we wanted to ask, how are these cells becoming binucleate? And we went and looked further into our live cell imaging, and we found possible mechanism. So I'll play this video multiple times because it's a little shocking at first. You're paying attention to this cell that's slingshotted across the screen, and as you watch it multiple times, you can see that this cell that moves all the way up here ends up rounding up, dividing, but it comes out with two nuclei. So this cell is an example of a cell that has failed cytokinesis. And I'll play that a few more times as I explain that binucleate cells in a population can arise from either cytokinesis failure, as I'm showing you here, or they can arise from mitotic slippage, which we do see some examples of cells, slipping out of mitosis before they have attempted cytokinesis, or cell fusion. We don't see any examples of cell fusion in our live cell imaging, but we do see high level or matching proportions of cells failing cytokinesis. And so here I show you a quantification of, the yellow bars are showing the quantification when you look at fixed cell analysis. So from those graphs I showed you earlier, I just pulled the data and I show that in control cells, when you look by fixed cell analysis, you don't see very many binucleates. You see more in the treated cells. And then when you look at the quantification from the live cell imaging and you quantify how much cytokinesis failure you see, we haven't seen any evidence of cytokinesis failure in the control live cell videos, but we do see a % of cells fail cytokinesis when we watch the treated cells in live cell imaging. And these numbers should be relatively equal because one leads to the next. So these results together have caused us to propose that cytokinesis failure is one of the main mechanisms that's leading to a binucleate phenotype from silver nanoparticle exposure. So in conclusion, we have shown that silver nanoparticle exposure does in fact reduce the viability of cell populations, which is in support with the previous literature, that it does in fact cause cells to die and to decrease viability of populations. But through our method using live and fixed cell microscopy, we were able to identify a new phenotype, something that has not been observed yet from silver nanoparticle exposure, and we were able to link it to a mechanism, in this case to cytokinesis failure. Now, you may be thinking, what's so important about having binucleates in your cell population even when they're at such small percentages that I showed you? But the thing is that binucleate cells are tetraploid. They have twice the amount of DNA that they should, and it has been previously shown that tetraploidy is a promoter of tumorigenesis. So the fact that we have binucleate cells arising in the population is a fairly important phenotype to be aware of. I also hope that in this experimental process I've shown that we can use my cell division based single cell microscopy approach as an important tool in understanding in vitro toxicology. And I say that as because we were able to identify a rare but threatening event while also highlighting the mechanism of toxicity. And with that, I would like to just quickly thank my lab, everyone in my lab, some collaborators, as well as my funding sources and my institution. And I thank you guys for listening, and I would love to hear any questions that you guys have.
Great. Thanks very much, Ellen. We have one question here so far. Aggregation seems to be an issue with nanoparticles. Is this an issue in your model system? And what is the cell cycle duration of your cells, and what impact do these particles have as the function of dose on duration? Okay, he has a lot of questions, so I'll go with those two first, and then we'll...
Okay. Yeah, so the first question on aggregation is, I've definitely read a lot of literature about people who have investigated aggregation of silver nanoparticles, specifically in cell culture systems, and I definitely think it's something that's happening, but I don't think it's something that I suppose is a negative effect on my experiments. I think that aggregation is a very natural experience for silver nanoparticles, kind of regardless of what system, whether you put it in fresh water, salt water, cell culture media, just pure water. It's something that is happening. It's a predominant behavior of silver nanoparticles, and so I think it actually adds to the realisticness of the experiments that I'm doing, that our body is more likely not going to be exposed to really small, stable nanoparticles. We as individuals are more likely going to be exposed to aggregates, or agglomeration will more likely happen in our bodies. So I think it's something that actually works well with my experimental model. And then as far as the duration question goes, I haven't specifically looked into how the entire cell cycle is changing in duration. So RPE cells have approximately a 24-hour turnover rate, so they are dividing about once a day, and so they go through, that would mean they go through the cell cycle in approximately 24 hours, and I haven't done any experiments to look at if, for instance, they stay in S phase longer, or they're in the entire cell cycle longer, maybe in G1 longer or something, so I haven't done those experiments.
Okay. Two more related questions. Let's see here. Can you relate the number of ingested particles to the mitotic delay? And go ahead and answer that one, because I think the second one.
I think that you could relate how many particles, oh, you mean ingested, like engulfed in my cells, not like what we would ingest as humans.
Yeah.
I think you could try. I don't think the method that I have is, at least I don't think it would be super able to do with microscopy, but I am in the process of doing some experiments where I'm trying to look at about how much silver is in the cells, and so I'm kind of trying to do those experiments right now. I'm looking to do some mass spec on how much silver is in the cell populations, but it would be very difficult at this point to do it for individual cells. Right now I'm just going to be looking at how much silver is in the cell population, knowing how many cells are in that population after silver nanoparticle treatment. But that's something to be looking forward to, because I am doing those experiments right now.
And then finally, how does the lowest active in vitro dose for your binucleation effect relate to likely in vivo exposures?
Yeah, so that's a really, really important question, because kind of anyone who's doing in vitro toxicology work knows that we need, there's certain levels of realism or to what we're doing. And so with my concentrations, I think that they represent kind of two possibilities of what we could be exposed to. I think that the higher concentrations that I use are pretty extreme. They're very high, and I don't think that that's concentrations that we as everyday people would be exposed to through using products that have silver nanoparticles in them. But I do think that it's an important concentration for people who actively work in manufacturing of nanoparticles and are making these products and are making nanopowder. I feel like they have a much higher risk of exposure to higher concentrations. And then the lower concentrations that I'm using, I feel like are more appropriate for what we would be exposed to, but there's still not a lot of literature out there on how much is actually being exposed in the environment, how many nanoparticles. I've been reading some papers recently trying to predict, but it's not an easy estimate to actually know how much we're being exposed to. So it's kind of all, at this point, guesswork.
Okay. I wonder if you could, I know that bandages are one of the uses. I wonder if you could figure out, people who are using bandages with silver nanoparticles in them, whether that is a measurable.
Yeah, there's a couple interesting ways you could do it. We've discussed possibly trying to incorporate materials into my cell culture model and actually having the material release the nanoparticles in direct kind of exposure to the cells. So we have talked about that as a method. And then there's also just the need for like an epidemiologist to come around, and maybe someone's doing this right now, but to understand what we actually do have. So people who have used bandages, what exposure do they have in their bodies? There's papers like that on other types of chemicals and nanoparticles, but we don't really see that kind of longitudinal study done with silver yet.
Okay. So we have a note, actually, instead of a question, from Stephen Derdinger. And he says there's interesting literature around failed cytokinesis. Check out Shi and King, S-H-I and King.
Okay. I possibly know the reference.
Okay, great.
Yeah.
I don't see any more questions. Reminder, if you have a question, you can type it in the question box, or you can raise your hand if I misstate your question or you have a follow-up or anything like that, and I can unmute you. And I think we're about out of time. I was wondering really quickly if you could, I know you're choosing silver nanoparticles for these experiments, but I wondered if the model that you've created is relevant to other types of nanomaterials.
Yeah, and that's definitely a really important question, and kind of a basis of my whole project is I really do want to develop an approach that can be used for all types of kind of chemical exposure, not even just nanomaterials. And so I think that the findings I found could be potentially very unique to my system, to the nanoparticles I'm interested in, but I think the approach of how I'm looking at responses and what I'm focusing on as a perturbation, like a system such as mitosis, is what's really important and what can be applied to other nanoparticles, other chemicals. And so what I really hope to do is use this similar approach, the same technique, with microscopy and single-cell mitotic analysis to understand how other nanoparticles affect cells, or other chemicals affect cells. So that's kind of in the works for kind of a validation step for my approach. Yeah.
Great. Okay, well, I would like to thank both of our presenters, Ellen Garcia and Emma Bowers. I'd like to thank you, the audience, for your attention. I'm sorry. There's one more question. Let's see here. Just a thank you, looks like. Thanks, Christy, for arranging this, even if it was an excellent program and an excellent discussion. Great. So thanks, everybody. A reminder that members can access the recording on the ASCCT website in the member section. And I'll just sign off with that. Thank you very much to both of you.